Assalamu alaikum friends. I hope so that you all will be fine. Friends, in this video I'm going to discuss about uronic acid pathway. So let's start our lecture. This uronic acid pathway, it is an alternative pathway for the oxidation of glucose. This pathway provides UDP, uridine diphosphate glucuronic acid. So another name, which is the active form of glucuronic acid, and the another name of uronic acid pathway is glucuronic acid pathway. This UDP glucuronic acid is very important in conjugation with steroid hormones, in conjugation with various drugs, in conjugation with bilirubin, and in synthesis of glucose aminoglycans. Okay, so friends, now coming toward the main pathway of this uronic acid pathway, various steps. Okay, so this glucose glucose by action of glucokinase enzyme ATP, it converts into ADP and inorganic phosphate. This glucose, it converts into glucose 6 phosphate. In next reaction, by action of mutase enzyme, this glucose 6 phosphate, it converts into glucose 1 phosphate. In next reaction, by action of UDP, glucose pyrophosphorylase enzyme, okay, with the help of this enzyme, it converts into, okay, here is UTP. There is removal of 1 phosphate from UTP and 1 phosphate from glucose 1 phosphate in the form of PPI, that is pyrophosphate, and it converts into uridine diphosphate glucose. Okay, friends, in next reaction, by action of UDP glucose dehydrogenase enzyme, and here two molecules of NAD plus, they convert into two molecules of NADH and H plus, and there is formation of UDP glucuronic acid, which is the active form of glucuronic acid. Okay, so there is a formation of UDP glucuronic acid. I am repeating, this glucose by action of glucokinase enzyme, it converts into glucose 6 phosphate, and then by action of mutase enzyme, it converts into glucose 1 phosphate. This mutase enzyme is responsible for the shifting of phosphate group from carbon number 6 to the carbon number 1. With the help of UDP glucose pyrophosphorylase enzyme, there is formation of UDP glucose, and here there is a removal of phosphate group from here and phosphate group from here, and collectively they release in the form of PPI, that is pyrophosphate, and there is formation of UDP glucose. In next reaction, by action of UDP glucose dehydrogenase enzyme, and NAD plus converts into NADH and H plus, there are two molecules. Okay, two molecule of NADH and two molecule of H plus, and there is a formation of UDP glucuronic acid. Okay, now next reaction, this UDP glucuronic acid, I am writing here. Okay, this UDP glucuronic acid, UDP glucuronic acid, by action of UDP glucuronidase enzyme, UDP glucuronidase enzyme, there is a removal of UDP, uridine diphosphate, and there is a formation of D-glucuronic acid. Okay. Then in next reaction, by action of glucuronate reductase enzyme, and there is NADPH and H plus, they lead to the formation of NADPH and H plus, they lead to the formation of NADP plus, and there is a formation of L-gulonic acid. Okay, L-gulonic acid is very important. This L-gulonic acid, it undergoes two pathways. Okay, by the removal of water molecule, this L-gulonic acid, it converts into L-gulonolactone. Okay, it converts into the L-gulonolactone, and in next reaction, this L-gulonolactone, by action of gulonolactone oxidase enzyme, gulonolactone oxidase enzyme, it converts into 2-keto L-gulonolactone. Okay, in next reaction, by action of again gulonolactone oxidase enzyme, there is a formation of ascorbic acid. This ascorbic acid, it is synthesized only in lower animals. It does not synthesize in mammals. I am repeating. This ascorbic acid, it is only synthesized in lower animals. It does not synthesize in humans due to deficiency of these enzymes that are gulonolactone oxidase enzyme. Humans can't synthesize ascorbic acid due to deficiency of gulonolactone oxidase enzyme. Okay. Now this L-gulonic acid, by action of gluconate oxidase enzyme, and there is NAD plus, it converts into NADH and H plus, and there is formation of, and there is a formation of 3-keto L-gluconate. Okay, 3-keto L-gluconate. In next reaction, by removal of carbon dioxide, it converts into the L-xylulose. Okay, it converts into the L-xylulose. जगह की तंगी की वजह से मैं यहाँ पर कुछ रिएक्शन लिख रहा हूँ यहाँ पर. This L-xylulose, by action of xylitol dehydrogenase enzyme, xylitol dehydrogenase enzyme, and there is a conversion of NADPH and H plus into NADP plus, and there is formation of xylitol. In next reaction, by action of Xylulose reductase enzyme, it forms, there is a for here NAD plus, it converts into NADH and H plus, there is formation of xylulose. And in next reaction, this xylulose by action of xylulose kinase enzyme, it converts into the xylulose 5 phosphate. This xylulose 5 phosphate, it is used in HMP shunt pathway. It is used in this xylulose 5 phosphate, it is used in HMP shunt pathway. Okay, so friend, this is the uronic acid pathway. This is UDP glucuronic acid by action of UDP glucuronidase enzyme, and there is a removal of UDP, that is uridyl diphosphate, and it leads to the formation of D-glucuronic acid. In next reaction by action of glucuronate reductase enzyme, and here NADPH and H plus, they are utilized and they lead to the formation of NADP plus, and there is a formation of L-gulonic acid. This undergoes two pathway, and here L-gulonic acid is removal of water molecule and there is formation of L-gulonolactone. L-gulonolactone. In next reaction by action of gulonolactone oxidase enzyme, there is a formation of 2-keto L-gulonolactone, and again with the help of this enzyme it converts into ascorbic acid. Remember that ascorbic acid is a vitamin, gets synthesized. Vitamin, it is a vitamin C. It only synthesized in lower animals. It is not synthesized in lower animals. It is only synthesized in mammals. In humans, it is does not synthesize in mammals. Okay. This L-gulonic acid by action of gulonate oxidase enzyme NAD plus converts into NADH and plus. There is a formation of 3-keto L-gulonate, and by removal of carbon dioxide, there is a formation of L-xylulose. L-xylulose. This xylulose by action of xylitol dehydrogenase enzyme, then NADPH plus, they converts into NADP plus. There is a formation of xylitol. With the help of xylulose reductase enzyme, the NAD plus, it converts into NADH and plus, and there is a formation of xylulose. And with the help of xylulose kinase enzyme, it converts into xylulose 5 phosphate, which is used in HMP shunt pathway. Xylulose, it is a 5 carbon atom keto sugar. Okay. Now, due to deficiency of these enzymes, humans can't synthesize ascorbic acid. Now, due to deficiency of these enzymes, that is xylitol dehydrogenase enzyme and xylulose reductase enzyme, there is that it leads to disorder, that is essential pentoseuria. Okay, and the deficient, this is a clinical significance due to deficiency of xylitol dehydrogenase enzyme and xylulose reductase, it lead to disorder, and that is pentoseuria, mean the excretion of xylulose in urine. Xylulose, it is a 5 carbon atom containing keto sugar. Okay, there are some drugs like aminopyrin and barbiturate that induce this pathway and they lead to the pentoseuria. Okay, this is inborn error. This is the inborn error metabolic disorder, and it is the member of Garrot's tetrad. This Garrot's tetrad, it includes albinism, L-captonuria, pentoseuria, and cystinuria. Okay, so this is the clinical significance point of view of uronic acid pathway, that deficiency of xylitol dehydrogenase and xylulose reductase, it leads to the pentoseuria. and it is an inborn and a metabolic disorder. There are some drugs that are iminopen barbiturate that induce this pathway and leads to the pentoseuria. It is inborn and metabolic disorder and is a member of Garrot's tetrad that include albinism, alkaptonuria, pentoseuria, cystinuria. This pentoseuria, it is not harm for our body. So friend, this was all about uronic acid pathway, the various steps and clinical significance. Keep watching and thank you.